உணவுத்துறை அதிகாரிகள்,
உணவு பாதுகாப்புத்துறை அதிகாரி,
ஃபுட் சேஃப்டி பிருந்தால்,
உணவு பாதுகாப்புத்துறை,
ஃபுட் சேஃப்டி அதிகாరులు,
ఫుడ్ సేఫ్టీ.
Welcome to Food Catalyst. This is Venkadesh and we are going to talk about foodborne disease. Without wasting any time, let's get into the topic. Foodborne disease. The term foodborne disease actually means an illness or disease which is caused by the foodborne pathogens. Okay, basically we are consuming a food which contains microorganisms and we are consuming the food and illness is occurring because of the microorganisms present in the food. Then what is foodborne infection? See, the foodborne infection refers to the microorganism which is present in the food and we are consuming the food and our body's reaction to the presence of microorganisms or the metabolites. That is the foodborne infection. See, the foodborne diseases can be further classified into invasive infections, intoxication and toxico-infection. Invasive means spread quickly or aggressively. Okay, it spread quickly or aggressively. So in this invasive infection there are two types. In one type where the bacteria uses the food as a growth medium, okay, that is culture medium, and the bacteria cannot use the food as a growth medium where the food does not support growth. So there are two types of invasive infection, and intoxication is also called as food poisoning. Okay, the term intoxication is called as food poisoning because in the food poisoning we are consuming the toxins produced by the microorganisms. So that's why it's food poisoning. And toxico-infection is a combination of these two, invasive infections and intoxication. And food poisoning can be further classified into chemical poisoning and toxicant poisoning. Under the toxicant poisoning, we have naturally occurring toxic substances or toxic substances produced by the microorganisms themselves. It can be either produced from the mold, which is mycotoxin, bacterial toxin and algal toxin. Under the bacterial toxin is further classified into endotoxins and exotoxins. And see, this food poisoning term is actually vaguely used. It is very loosely used in India or in all over the world because this term is actually used for both the invasive infection and intoxication. They used to consider food poisoning as a general term. But actually what? The food poisoning is only meant intoxication. So the further classification of foodborne diseases is clearly shows that it is further divided into food poisoning and infection. So in this case, the food poisoning is only the result of chemical poisoning or toxicant poisoning. Okay, remember this word. Let's talk about these three terms in detail. What is an invasive infection? It means we are consuming the food which contains the bacteria and the bacteria is viable. Viable means the viable cells are the cells which is potential to penetrate into the epithelial cells of gastrointestinal tract and multiply, which simply means it can do the function of invasive. It can come into the body and it can grow and multiply. If some bacteria can do it, then it's viable. If the bacteria is dead or it cannot able to do the viable function, then that's an inviable or non-viable bacteria. Okay, so this invasive infection is basically due to the entrance of the bacteria, okay, into our body, and the body is reacting to the bacteria as an infection, which we have seen before. Okay, there are two types of infection. First one is the food does not support the growth. There are some examples: tuberculosis, diphtheria, dysentery, typhoid fever, brucella, cholera, Q fever and infectious hepatitis and all. So these examples which I am going to give you is really important because see, here the food is not supported as a growth medium. So these microorganisms are barely present. It present less than like 1000 cells or something like that. But next para we can see Salmonella, Vibrio and then E.COLI and Shigella is present in a major manner. Why? Because the food is acting as a growth medium, so it can produce multiple viable cells. So it can reach like 10 power 6 to 10 power 10 and all. So that's why the second medium is the most medium which we can see in the outbreaks. So majority of the foodborne outbreaks is caused because of the type 2 bacteria, okay, which is more explosive than the first bacteria. I have taken the 2016 outbreak disease record. You can see the food poisoning is the top disease causing disease outbreaks happened along with the measles. So see, as I told you, food poisoning is vaguely used. So this contains both the infection and the intoxication. Okay, so the food poisoning considered as a major disease outbreak. Then you can ask me whether how you are saying that only type 2 is responsible for the food poisoning, right? Let's take the table of all the reported outbreaks happened. As you can see, all the highlighted ones are type 2 where the food is acting as a growth medium. So you can clearly see that the reported foodborne outbreaks happened in 1980 to 2016 is basically because of the type 2 diseases. Okay, so you need to take care of that. So let's move on further. What is a intoxication? Okay, intoxication or poisoning, food poisoning, it's basically we are consuming the toxins produced by the microorganisms, which is previously produced by the microorganisms, plants or animals or chemicals present in the food. Okay, then you can ask me whether we need viable microorganisms or not. We don't need a viable microorganisms in the food. Just the toxins is enough to make the food poisoning. Simple. It can cause gastrointestinal and systemic disorder. Let me tell you a point clearly. Why it can cause gastrointestinal and systemic disorder? See, in order to reach an intestine, it has to go through the pH range of our intestine, right? And the temperature is also high. These toxins are mostly stable to that heat, but microorganisms are not. That's why most of the invasive infections will cause infections in the body. It cannot able to reach the gastrointestinal or intestine. If they have to reach the intestine, they have to be in a very high number, like 10 power 4 to 10 power 10. Then what happens is, if they are in the very high number, then this bacteria can die and leave a few bacterial strains in the intestine. That bacterial strains can further cause disease. So two things. One, this invasive infection bacteria should be in a really high number, or it has to be protected through food. So the food will act as a barrier and it will carry the bacteria to the intestine. Okay, so that's the difference between the intoxication and the invasive infection. Okay, now toxico-infection, it's a combination of both. First, what happens is the entrance of the viable bacteria into our body, and once it enter our body, it will release a toxin. Okay, so it will enter our body and release a toxin, right? It will release a toxin only in the intestinal area. So how it releases a toxin, I'll tell you that. First of all, this bacteria, once it enter into the body, it has to be really large number of viable pathogenic bacteria to reach the gastrointestinal tract and cause gastroenteritis. And how it released into our body is because of the sporulation or lysis of the bacterial cells. Okay, so and it will release a toxin. So it is basically doing the function of the first and second types of bacteria. Okay, that's it. So we can say it's a combination of about two types. So it has to be viable and has to release a toxin. Okay, now let's talk about the pathogens and diseases and how we should study it. For exam perspective, you need to know the characteristics of bacteria, you need to know the disease, infective hosts and symptoms, and sources and how it is associated with the food, then prevention control, finally isolation and identification. If we have studied this for the pathogens and diseases and toxins, you are good to go for all the examinations. Okay, so see if you are studying for CFSO, TO or any other FSSAI or FSO related examination or GATE examinations or CFTRI examinations or any other entrance examination, even patent examinations, patent examiners, you can study this topic. Okay, so let's take an example of how to study this. CAMPYLOBACTER. Okay, this is the family is actually Campylobacter, and the example is Campylobacter jejuni, E.COLI and Lardis. So it is a non-spore forming, catalase positive and then oxidase positive bacteria, and it's also a gram negative bacteria. Okay, then see, I will explain this, what is this, in another class where I will explain the basics of microorganisms. Now this class we are just going to talk about in general. Okay, so this is the characteristics of Campylobacter. First is non-spore, catalase positive, oxidase positive and gram negative. It is small and spirally curved long rods. Okay, it's a rod bacteria. and it's actually motile, means it has a flagellum at both ends, so it can move from one place to another, and it's actually showing a rapid darting motion while moving. Okay, in a chemical perspective, it cannot ferment carbohydrate, it cannot oxidize carbohydrate, or gelatin are not hydrolyzed, which means it will not produce any slimes. Okay, optimum temperature is 42 to 45, means it will mostly cause diseases in the summer. Why? Because it cannot grow below 30 degrees Celsius, means in the winter time where the optimum temperature is less than 30, it cannot able to grow, so it has to be in the range of 30 to 45. So that's why the diseases are more in summer, and it cannot survive the pasteurization temperature also. So if you cook it really good, then the bacteria will die. There are other sensitive factors too along with this cooking temperature, that is drying, NaCl greater than 2.5%, is salt greater than 2.5%, and then pH lower than 5.5. See, this is really simple. If you are just maintaining a pH below 5.5, it will not grow. Then what is an optimum? It's 6 to 8. This proves that it is killed by the gastric acid and is protected by the food materials. Okay, it is sensitive to oxygen, but it is a aerophilic, but it is a sensitive to oxygen. Why? It is called as microaerophilic, means if oxygen is present in really high number, okay, if oxygen is present really high number, then it cannot able to survive. Okay, then it cannot able to survive. Then even freezing is also it's also a sensitive factor, because we know that it will not grow below 30 degrees Celsius. Then gamma radiation. Disinfection is also a sensitive factor. Okay, let's move on to the disease and dosage. Disease is actually Campylobacteriosis, and dosage is 10 power 4 bacteria should be there per gram. Okay, this Campylobacteriosis disease can be divided into two types. One is extra-intestinal infection and gastrointestinal infection. Extra-intestinal infection is actually the infection once it enter into our body. Okay, this is when it enter into gastrointestinal tract, the second type of disease, and it's called as acute enterocolitis. I'll explain that later. Let me tell you how it will affect the gastrointestinal entities. Okay, if the food contain 10 power 4 Campylobacter jejuni, with the production of the food, it will go against the acidity, so it will not affect through the acidity, and it will reach the intestinal tract. Once the bacteria has reached the intestinal tract, it will produce something called invasive factors. This invasive factors is a small bacteria. It will go and utter the intestinal epithelial cells. Okay, it will utter the epithelial cells. How it can utter? We know it has a flagella, but along with the flagella, it will also produce something called antigen, superficial antigen, PEB1. Using the flagella and antigen, it can able to utter into the epithelial cells. Okay, by adhering to the epithelial cells, it will cause the gastrointestinal symptoms. But what about the extraintestinal function? What happens is, same invasive factors, it will go into the intestinal mucosa. It need not to go into epithelial cells and all. Just the outer layer mucosa it can enter, and it will die in the gut epithelial mucosa. Once it has reached the gut epithelial mucosa layer, it will die, and it will produce something called enterotoxin. Okay, after dying, it will release the enterotoxin. This enterotoxin will stimulate something called adenine cyclase. Okay, this adenine cyclase is really important because it's useful for the muscle contraction and everything, right? And it will further produce the cytophatic and endotoxin. So basically, it produces all three: enterotoxin, cytotoxin, and endotoxin. See, this bacteria cannot survive the heat, but this enterotoxin can survive acid and heat. Okay, so that's why after producing the enterotoxin, its job is done actually. That's it. And see, in an exam perspective, this adenine cyclase can stimulate by only few bacteria. One is E.COLI, cholera, Bacillus cereus, and also this bacteria Campylobacter. And along with that, we can also say Clostridium botulinum can also do this function. Clostridium botulinum. Okay. So that's it. Now you know how the food and Campylobacter can finally affect the gastrointestinal or extraintestinal infection, right? Let's talk about symptoms. there are two types of symptoms. One is the headache, fever, profuse diarrhea symptoms with blood and because of with or without the blood diarrhea. Okay, and vomiting is less common, and we cannot die from this the Campylobacter, and some gastrointestinal infections also there, and the mortality rate is actually 5 to 10%, and the symptoms is usually as 2 to 6 days. There are some orthotic and nervous diseases also, but the major symptom is this one, which is mimicking the acute appendixes. See, people used to do appendix operation if they have the acute appendicitis, right? What happens is, since it can go and attach the large intestine epithelial cells, it can stimulate a kind of symptoms. Okay, since terminal ileum and cesium are often affected and causes the symptoms that mimics the acute appendixes. Okay, you can ask me, okay, what happens if somebody has been misunderstood with this acute appendixes? They will do an unnecessary surgery, which is not at all needed, because you don't have the acute appendixes. Thus, it just will give you the symptoms. Okay, this leads to unnecessary surgery. So this is one of the major symptoms of Campylobacter. Then sources. So we know it is present in the intestine, right? And it has a high temperature. So it can survive warm-blooded animals' intestine. Okay, it is majorly present in the warm-blooded animals' intestine. So it is called as zoonotic, since it is present in the animals. Okay, and this makes this animal as a reservoir. Okay, this it's a zoonotic. So most of the animals can be act as a reservoirs for this bacteria. Okay, we should only focus on the commercial animals, because that's what actually matters. Even wild animals can affected through this Campylobacter, but we only focus on the commercial animals like rabbit, rodent, sheep, horses, and domestic pets. Okay, the fecal matter of this commercial animals will linked with the surface water, vegetables, meat, eggs, clams, and mushrooms. It can somehow contaminate these foods, and by consuming these foods, we can affected with the Campylobacteriosis, right? This bacteria is called as enteric bacterium. Why? Because it transfers from one person to another person. Okay, this is an important point. It transfers from one person to another person. We can see the commercial outbreaks happened with milk, water, and seafood and egg. Okay, so this is the sources. Now let's talk about prevention of this bacteria. Prevention is basically simple. We have already seen few of them. Safe disposal of sewage, treatment of potable water, heat treatment of milk, and general hygienic process is needed, because it's linked with the feces, right? And we can heat treatment because we know it cannot survive pasteurization or cooking temperature, right? And we need to somehow stop the transmission of bacterium from the food producing animal to the final product. By doing this process, we can control the Campylobacter many ways, like contamination from farm pets to animals, and then good food hygiene also used to control it. If somebody has diarrhea, they should not work. So these are like basic symptoms. You can read it out. Okay, after give you these PDF, you can read it out. And one important thing is, it has to be cooked internally 77 to 80 degrees Celsius. The internal temperature of the meat should be 77 to 82. Then the bacteria will completely die out. Okay, then isolation and identification. So let me tell you simply how we can isolate and identify a specific bacteria. Not just this, any bacteria. In order to isolate a bacteria from a group of bacteria, we need to give the specific bacteria which it likes to grow. For example, this bacteria will like to grow in a specific enrichment medium. For example, if you like only chocolate, where your friends likes pizzas or any other food items, if you give you the chocolate, you will come immediately. But since your friends likes pizzas or other items, they will not agree to come with you to eat or go out or something like that. So that's why we are enriching the Campylobacter with the selective enrichment broth. In this case, we are using polymyxin and trimethoprim. Okay, but there is one problem into it. It is sensitive to oxygen, so you cannot do it in a normal condition. You need to have a microaerophilic condition to do it, to do the isolation and identification. Okay, and we need to make the enrichment 24 to 48 hours and give 42 to 43 degrees Celsius, because we know it's a optimum temperature. Then we need to do the streaking method and selective plating method, and to identify the colonies we need to do motility, morphological test, biochemical test. Then to identify specific serotypes we need to do phage typing and molecular typing and all. That's it. That's how we have to isolate and identify. Now let's talk about Clostridium. Clostridium, we have two important, one is botulinum and periphenes. Botulinum is actually intoxicant and periphenes is actually toxico-infection. We can say infection basically, but intoxication. Even toxin can be produced from periphenes. So this is only intoxication. So in general, it's a gram-positive bacteria, motile. It has trichous flagella. It's motile. It can move from one place to another, and it has a peritrichous flagella. Obligate anophilic rods. Endospores is present. It can produce spores, okay, but it cannot germinate the spores if you are giving nitrogen 250 ppm. Important point. And it can produce toxin. That toxin is neurotoxin. So basically, Clostridium botulinum toxins are divided into three different types of groups: human toxin, fish toxin, birds and animal toxin. Human toxin is A, B, E and F. Fish toxin is just E. Bird toxin is C and D. Okay. See, the human toxins is called as proteolytic strain, and it's a mesophilic. The strains A, B, E and F, not E, just A, B and F, they are grouped into a single category called proteolytic strain group 1. This E is a non-proteolytic strain, psychotrophic, and the birds E and D is actually a group 3 of non-proteolytic strain. It has both psychrophilic and mesophilic strains are. So this is how we have to divide the Clostridium botulinum into three different group of strains, and the pH is actually 4.7 to 8.9. This is a very important question because it cannot survive less than 4.7 pH. So optimum pH is 6.5. If you are having a acidic food which is less than 4.7, then you can simply skip this microorganism because it will not survive. Water activity is 0.93 is also important, and it can survive the salt moderately because it 5.5% of NaCl is easily able to survive it. Now comes the question, what is a proteolytic, right? Proteolytic simply means it can able to break down and degrade the protein. Okay, let me explain it in a simpler way. This proteolytic bacteria has an ability or able to produce enzyme such as proteases and proteinases, which can break down the protein into smaller peptide and amino acid. That is okay, but why it is important for a bacterial strains? See, this proteolytic strains which can easily utilize the protein present in the environment. Okay, if the protein is present in the environment, it can utilize the protein and it will break down the protein and it will take the nutrition out of it. So it can grow easily. But in such cases, in non-proteolytic bacteria, it is lacked with the ability to produce the proteases. Then it cannot utilize the protein. Then what it will do? It will do the alternative mechanisms such as utilization of carbohydrates or organic matter. So it does not require the proteolytic activity. Other bacterial strains. Example of such strains is present in the lactic acid bacteria. Okay, proteolytic bacteria which we can see it's present in the Clostridium. Okay, so and also in the Clostridium we have non-proteolytic strains also, but this non-proteolytic strains is majorly present in the lactic acid bacteria. Okay, this bacteria is often ferment carbohydrates without need of the extensive protein degradation, right? Okay, now here comes the second question, nature and association with food. One important MCQ is present in the soil, and freshwater sediments of lakes and coastal water. From the soil to it will transfer into the intestinal tract of animals and fishes. From that we will consume in a fish, meat, chicken or infant foods. It also come from the honey, so it can also affect the infant. So it can transfer from here to here to here. In fruits and vegetables, actually the normal Clostridium botulinum will not transfer to fruits and vegetables, but the spores which is only able to produce in the soil can go to the fruits and vegetables, and it can leads to this further will be grown in the low acid canned food. If the canned food is low acid, then it will act as a growth medium, and insufficient heat and without oxygen it has act as a growth medium. That's why MAP processing foods can also lead to Clostridium botulinum. Minimally processed food is also leads to Clostridium botulinum. RTE, ready to eat foods, also leads to Clostridium botulinum. Why? Because we are giving a condition that it can grow. That's why less or irregularly processed food leads to this Clostridium botulinum disease, growth of this specific bacteria. What are the diseases? We have 5 different diseases. First is a classical foodborne botulism, which is occurring because of the neurotoxin called as BONT, botulism neurotoxin. This is commonly seen in the improper processed food. Second type of diseases is actually caused from the infant botulism. It is an infection. It's an infection. Important point, infection. Why? Because this bacteria will germinate and it will create a spores, and that spores can enter into the intestine. Why? Because spores can able to survive the pH and temperature of our intestine, and it will cause a disease called floppy baby syndrome. If the exact same process happened in adult, is called adult infectious botulism. Okay, wound botulism is similar to the infant botulism, because here also spore will germinate in the wound, but it will release a toxin in the end and into the deep wound. So that is wound botulism. Inhalation botulism is simply we are taking the botulism, even a 2 nanogram is able to kill a person. That's how dangerous it is. Dosage is actually 2 nanogram. So that's how dangerous it is. So we are injecting the botulism into an aerosols and releasing it to the environment. So it's actually bioterrorism, right? So that's why it is very, very rare, but it can use to kill other people also. Okay, so let's talk about the toxins. The toxins has been divided into 2 types based on where it is, how it is produced, and the site of action, where it is acting. Let's discuss the action only. See, the site of actions can be divided into enterotoxin, neurotoxin and cytotoxin. Under the enterotoxin, it is act in the enter region. What is entero or enteric? It is intestine. Okay, it is in the gastrointestinal tract. Example of such a toxin is cholera toxin and Shiga toxin. Under the neurotoxin, we have Clostridium botulinum, which will interfere with the nerve impulse, which we are seeing right now. And cytotoxin is simply a toxin which attacks its host cell itself. For example, if a toxin is attacking its host cell through enzymatic attack, then it's called a cytotoxin. Example is diphtheria toxin. Now you have understood the toxin. We'll study the enterotoxin and exotoxin later on, but these are the 3 important toxins. Then symptoms. First of all, let's understand the symptoms. First is within 18 to it's like a cycle. First, within to 18 to 36 hours, you know that you have the symptoms by consuming the neurotoxin of Clostridium botulinum. The symptoms will start at 18 to 36. Okay, it may be from the small vomiting or any other enteric symptoms. Then what happens, this toxin will come into our body and it will circulate through the entire body and small intestine. Then it will reach to the bloodstream. Okay, this is the second step. Then what happens, it will go and it will bind with the nerve muscle junction. Okay, because obviously it's a neurotoxin, and it will release the acetylcholine, which is essential for the muscle contraction. So you cannot use the muscle contraction because it blocks the release of acetylcholine. Then you will get the early gastrointestinal symptoms, pain, nausea, vomiting and all, followed by the neurological symptom, followed by the throat functions and normal mouth functions will be lost, followed by the muscle coordination will lost and respiratory impairment will be there, followed by the followed by the mortality or death. So this whole process lasts for 7 days. Okay, this whole process will last for 7 days. So this is how this specific symptoms of the bacteria will occur. And actually the mortality rate is differ from bacteria to bacteria, but it is majorly high in A, okay, compared to B and E. And the toxin lethality is highly toxic, because we have seen 2 mg is enough to do a bioterrorism, right? So as long as 1 mg per kg of the body, 1 mg per kg of the body is enough to kill a person. Okay, then it can also do genetic encoding, so it can alter our chromosomes and all. Preventions and control. There are many prevention mechanisms are there, but I am going to mention only few of them. First one is, we have to inactivate the bacteria before producing the toxin, simple. Inhibiting the bacterial growth, simple. And preventing the contamination from one food to another, okay, simple. Then home canning process, strictly hygienic procedures should be followed. Okay, we cannot do a false canning process like without giving the proper temperature and everything. Okay, then 12D concept. This 12D concept is 12-fold reducing of bacteria. Okay, so I will explain this 12D concepts later on in some other video. Okay, I'll explain this concept detailedly, but right now the 12D concept has been used. At least 7D concepts is needed, but 12D concept is needed for the specific Clostridium botulinum. Okay, then salting and smoking, storage conditions and preserving techniques, and then we have seen nitrate also, right? Preserving techniques, resistant, we are using the highly radiated resistant bacteria, like this radiation inactivation will be there, and then inhibition of the spores and germinations and all. Okay, then medium. Here for the isolation identification, we are using cooked meat medium as a growth medium for the bacteria to grow. And to create the same environment, we need to do it we need to do like without oxygen, right? That is the first one. And to eliminate the non-spore forming bacteria, heat it for 80 to 10 minutes, 80 degrees Celsius for 10 minutes. All the non-spore forming bacteria will be destroyed, only the spores will be there. And after doing an elimination process, keep it at 35 degrees Celsius for 7 days, because that's how the days we know that it can able to be there, right? It can able to survive that, right? And we can also do the some other checks. For example, we can check the characteristics of the colonies, and we can also check for the toxin, neurotoxin, right? Neurotoxin toxins will be there, and you can inject it into the mice and see when it is dying. Usually the death will happen for 48 hours. So if the mice has died in 48 hours, then you can know that it's because of the neurotoxin. So Clostridium botulinum can one of the responsible for the specific disease. Okay, now let's talk about the other two types of toxins. See, this is based on this two types of toxin, exotoxin and endotoxin, is divided based on the production, which I have told you before. Okay, let me explain it in a simpler way. Production. See, I have two toxins, exo and endo. Okay, exotoxin and endotoxin. Endo means endogenous toxin. Endo means endogenous toxin. Endo means endogenous toxin. So here the toxin is produced within the cell. Okay, when the cell is lysed, or when the cell is dead, or when the cell is removed, the toxin is released into outer environment. But in exotoxin, the cell will produce a toxin outside itself. Okay, it is actually a kind of like a production unit. Okay, where it will produce a toxin outside. It will continuously produce in a series of intervals. But in endotoxin, it will produce only once when the cell dies. Now you know the difference between exo and endotoxin. This is a simple difference. You can study the whole PDF if you want, but this is a simple explanation. Okay. Now one more point is it has to be, the cell of the bacteria has to be destroyed to release it. So it will released from the cell wall in the endotoxin. Okay. So that cell wall is actually made up of lipopolysaccharide, because only in that cell we can able to release a toxin. It is present in the gram-negative bacteria. Okay. So let's discuss this toxins further. Type of toxin is LPS and peptidoglycan, and in the exotoxin we have enterotoxin, cytotoxin and neurotoxin. We have already discussed what it is, and we can say it is pyrogenic, it is enteric, and then it is a cell associated endotoxin. Exotoxin is cell, extracellular and diffusible. Denatured by boiling? No, it is usually heat labile. Okay, and then you can read the whole things if you want. These are the important points. It is low specific actually, because it will just release at any point of time. When the cell is dead, it release. But this is highly specific, because it will release specifically for a specific type of the location which is present in it. That's why it is enterotoxin, cytotoxin and neurotoxin, right? Because it's highly specific. Simple. That's it. This is the explanation of endo and exotoxin. You can read it the whole page to understand more. And as I have mentioned, it is heat liable, it is inactivated by boiling. You know that already, I have explained it. Okay, now gram positive and gram negative bacteria, what it is and how we can differentiate. Before knowing gram negative and gram positive bacteria, what is a cell wall? Okay, like to understand the cell wall, let me tell you clearly, it is a structure layer, outer structure layer, which is tough, flexible and rigid, and it's the one which surrounding the cells. Okay, mostly bacterial cell walls will be there, but apart from that, most like prokaryotes, we have algae, fungi and eukaryotes is also there. And in plants, cell wall is present, but in the animal, cell wall is absent. So that is the cell wall. Cell wall contains cellulose and peptidoglycan. You know cellulose, but what is a peptidoglycan? Peptidoglycan is a series of 2 amino sugar and 4 amino acid. The sugar name is NAG and NAM. Okay, you have NAG, NAM, NAG and NAM. And in the NAM, it is attached with 4 amino acid, alanine, glutamine, L-lysine and D-alanine. Okay, it is called as tetrapeptide. So the NAG-NAM is attached with the tetrapeptide. Okay, 4 amino acid, it collectively called as a peptidoglycan. Now you know what is a peptidoglycan. Let's move further and understand the 2 different types of cell wall, gram positive and gram negative cell wall. First, let's understand the gram positive cell wall. In the lower layer, we have plasma membrane and integral proteins, followed by the periplasmic space. It acts as a cement. Okay, then we have the peptidoglycan layer. Peptidoglycan, I have already told you what is a peptidoglycan. It is a thick layer present in the gram positive cell wall. Since it is very thick, it prevents osmotic lysis. Okay, it has 2 types of cell attached to it. One is the tetrachoic and lipotetrachoic. Okay, the tetrachoic is the one which makes the cell stronger. Okay, and you can see the small strings out of it at the end. It is called a surface protein. It can act as an enzyme or adhesions to attach it. Okay, it is a 1 layer cell wall. Gram negative cell wall is actually 2 layer cell wall with outer membrane. Outer membrane is the lipopolysaccharide. Okay, you can see peptidoglycan here, but it's very much thinner than the previous one. So it's like 10 nanometer thin. Here it is 20 to 80. Here it is just 10. And you can also see reverse in the in the periplasmic space where it is very smaller, here the periplasmic space is bigger. And along with that we have a porine and O specified side chains. Okay, it is also in the outer membrane. Okay, see, as I told you, it is very much thick. It cannot allow anything to pass on. But here, here it has a lipoprotein and peptidoglycan in a very small chain, so it can regulate the lysine. Okay, because of the lipoprotein peptidoglycan, it can regulate the lysine. And I have told you, right, endotoxin will be reduced. The endotoxin will be reduced in the gram-negative cell wall, right? I have mentioned it where here, right? In the gram-negative. Okay, so basically from here only, from the destruction of this specific layer, it will be reduced. It will be released into the outer layer. So that's why the endotoxin released from the part here. Okay, now you understood the two different structure of the cell wall, where this point is important. It prevents the osmotic lysis, where it regulates the lysis. It will allow it to pass, it will not allow it to pass. That's it. Now let's understand the gram staining. See, basically this technique is used to differentiate the two large group of bacteria based on the cell wall. Who identified this first? Franz Christian Gram. In 1884, he is a Danish scientist. Okay, the process is actually simple with 4 steps. First, stained with the water soluble dye, crystal violet. Next, do the fixation, then decolorization, then counter stain. Let me explain. First, stain with the water soluble dye, which is crystal violet. Now this dye will both coat the gram negative and gram positive bacteria. Both are purple in color. Now I am adding iodine. What happened is, because of the thick peptidoglycan layer, which is on the outside of the cell wall, it allows the complex formation of crystal violet and the iodine. So here the iodine and crystal violet will be bond here, but here you can see it is present downside and it's really thin. So here the complex will not form in that layer. Only in the gram positive the complex will be formed. In the gram negative there is no complex formation. So next, decolorization step, where you use ethyl alcohol 95%, it will wash out the gram negative purple color, but gram positive purple color will be present because of the peptidoglycan. Okay, now what happens? We are using a counter stain to color the non-bonded gram negative bacteria, because gram positive is already made a complex here, right? So it's in purple color. Now the gram negative will be red or pale pink color. So this is the different gram positive and gram negative bacteria. Okay, done. Now we are done with the gram positive and gram negative bacteria. Let me give you some examples of what is a gram positive, gram negative bacteria. This is not in any books or anything. This is just my understanding, perception, that I can divide this gram negative into these different types, so you can easily study. Monas, we have Aeromonas, Plesiomonas, Ultramonas, Xanthomonas, Pseudomonas. Under bacteria we have Campylobacter, Acinetobacter, Guelphinobacter, Acinetobacter. So you can study it like this way. Okay, where P means psychrophilic, M means mesophilic, A means aerophilic, FA means facultative anaerobic. Sorry, A is aerobic and FA is facultative anaerobic. So this way you can study it. Under bacterium we have Flavobacterium and alkaligens, proteins, and Vibrio we have, and it's a facultative anaerobic mesophilic. Under spore forming we have Sulfotomaculum. This is the only spore forming bacteria. So it's actually anaerophilic and thermostable, because it's spore forming, right? It can produce H2S, hydrogen sulfide. Okay. Under Ricustets we have Coxella burnetii also, which is also gram negative bacteria, and it's actually non-motile, highly resistant to heat temperature, but pasteurization temperature it cannot survive. Coxella burnetii also can cause cattle fever in cattle, Q fever in humans. The simple. Here the important point is essential. Let's talk about this E. coli. First of all, this E.COLI, Theodor Escherichs was the first German scientist who has isolated the E.COLI in the children feces in 1885. And he called it bacterium E.COLI communi. Why? Because it's found in the colon. Colon. And E.COLI. Simple. That's how the name has born, I think. Then this is a very important question. It's established as a foodborne pathogen in 1971 only. Because while checking the imported cheese, they find E. coli. Then they got to know that okay, it can act as a foodborne pathogen. So in 1971, it is established as a foodborne pathogen. Then in 1996, in Scotland, a worst outbreak of E. coli has happened. Okay, they have mentioned as food poisoning, even though it can cause both infection and poison. They are still mentioning it food poisoning, but worst outbreak of E. coli happened, and there we get to know that it's a 5 group of virulent group is present. One is enteroaggregative E. coli, hemorrhagic, invasive, pathogenic and toxigenic. Okay, these are the different types. In this one, E. coli with a number of O157H7 is a predominant bovine origin. Okay, is the bacteria which you are mostly seen in many like laboratories and everything, because they will keep this as a standard bacteria, and it can easily give the off order, which is Borne order, which used to used as an identification method also. Okay, in the specific hemorrhagic E. coli, it can produce cytotoxin which is similar to Shiga or Verotoxin. Okay, it can ask as an MCQ question, so I mentioned it. Under gram-positive bacteria, we have Koki, Micrococcus, Staphylococcus, Streptococcus, Enterococcus, Lactobacillus. And heterofermentation and homofermentation is also I have mentioned here and there, so you can study it. And under the rods, Koki and rods, under the rods we have endospore forming rods, rods without spore forming, irregular rods with non-spore forming. Okay, and we have also, this is also Koki, Sarsinia is also Koki, Lichenostock is also Koki. And I have divided this into Arabic, as you can see, like the division is there. Then let's talk about every single pathogenic organisms, invasive organisms, and then like toxic infection organism. Before that, you need to know a prerequisite. What is an enteric symptom? The symptom enteric means gastrointestinal symptoms like diarrhea, abdominal pain, nausea, and vomiting and cramps. Because this enteric is also mean intestine and digestive tract. Non-gastric symptom means not related to stomach, like fever, fatigue and headache. Systematic symptom means fever, chills, malaise and muscle ache. Malaise means like the tiredness. The systematic symptoms means it affect the entire body, not just the intestinal tract, not just unrelated stomach like fever or fatigue. It affect the entire body. Systematic means. Okay, let's talk about one by one diseases. Like what's the disease, what is the microorganism, what is the dose, sources, food and symptom. First is brucellosis, and the species is Brucella, obviously. In 3 to 21 days, greater than 500 bacteria, it will cause a disease. Cattle, poultry, sheep, raw milk, dairy, it will transfer from cattle to raw milk, dairy, poultry, and it can cause both enteric and non-gastric. Next is CAMPYLOBACTER, we have seen, is a jejuni, 2 to 5 days, 10 power 4 viable, we have seen it. Cattle, poultry, sheep and raw milk, and then it transfer from here to uncooked meat, boiled chicken, hearty foods, and it will cause diarrhea, fever, nausea and vomiting. I haven't mentioned one symptoms. If you are like watching this video like above 45 minutes or so, I'll ask you one question. What is the one symptom which I haven't mentioned here? It's an important symptom. You have 5 seconds to write it in the comment section. You can pause the video and write, and I'll tell you the answer. 1 and 2, 3, 4 and 5. And the answer is it can mimic the acute appendixes, right? It's an important symptom. Having mentioned it so that I can ask you the question. Okay, so next is hemorrhage E. coli. Okay, see, this E. coli is the one which makes them to establish as a foodborne pathogen. 2 to 21 days, 100 to 10 to 100 viable bacteria is needed. Okay, cattle, poultry, sheep and raw milk, it is the sources, and it transfer from here to uncooked meat, raw milk, juice and fruits and vegetables. and it can cause diarrhea, fever, nausea, and vomiting. In the invasive E. coli, it is used as an indicator of fecal contamination because it's invasive. It's basically from human to like cheese or any other kind of things. Okay, because of the fecal contamination, it may transfer. And 3 to 24 hours, you can identify this, and pathogen is 200 to 500 viable is enough. And it can cause moderate dehydration. Both of this E. coli can be identified to ELISA test. Okay, through ELISA test. Listeriosis is caused by Listeria monocytogenes. It's 1 to 7 days and greater than 100 viable cells needed. Soil, water, silage, decayed vegetables is important, is responsible for transferring into soft cheese and milk, and it can cause nausea, diarrhea, and then streptomycia and meningitis. Okay, then salmonellosis, important pathogenic bacteria, because that is caused by Salmonella species. 12 to 48 hours we can see 10 power. See, quick example. In the very first 10 minutes of the video, I explained what is the two types of infective disease, right? One is the one which can food is acting as a growth medium. Example, this here, food is not acting as a growth medium. That's why very less viable pathogen is there. Here more pathogen is needed because it has to travel throughout the food. Herely the typhoid fever, the food is merely acting as a carrier. That's why very less viable bacteria is there. Here it need a huge number of bacteria. It's a quick example. Typhoid fever, and then here it's Salmonella typhi, and here is Salmonella species. Okay, all other Salmonella species. Here it's Salmonella typhi, the specific bacteria. And it can transfer from humans to milk, meat, and poultry, and it can cause enteric and systematic symptoms. Shigellosis. Who understands the disease? First of all, shigellosis is actually Kiyoshi Shiga in 1898, and he has named this as dysentery, Shigella dysentery. 1 to 7 days, 10 to 200 viable pathogens is enough. And human and water, it transfer from human and water to potato salads and waters. And diarrhea, it can cause water and bloody diarrhea. Dysentery, diarrhea, easily you can understand it. Yersiniosis is caused by Yersinia enterocolitica. 24 to 36 hours and 10 power 4 to 7 viable cells needed, from water, poultry, wild animals to milk and poultry, and moderate diarrhea to chronic diarrhea. Okay, shigellosis and yersiniosis is basically diarrhea. Simple. Then vibriosis. This vibriosis is really important. Why? Because it's a halophilic. It's a halophilic, and it's basically attack in the sea water, marine or marine life, basically aquatic life. Okay, so that's why it can affect seafood, shellfish and oysters. Then let me tell you an important point. In the recent outbreaks in Tamil Nadu, 8th standard girls died because of consuming I think it's shawarma, and they have find out that it has a vibriosis present inside the shawarma. You might be wondered how the hell vibriosis can cause can come in the shawarma, because it's not listed here, right? Here is a twist. What happened is, he cooked the shawarma, and if any shawarma leftover is there, he put it in the refrigerator. Along with this cooked shawarma, it also contains fish and other materials as well. So it transferred from the raw fish to the chicken to the cooked shawarma, and that's how it has been transferred. So you can see this is a halophilic, so it can grow in the chilled environment, right? So that's how it transferred into the shawarma. And because of consuming this, you can see the symptom is abdominal pain and fever and diarrhea. Even though symptoms is this, it leads to death. That child is actually dead. Why? Because of the ideological behavior that they can put everything in the same thing. No, it not works like that. And this will lead to also a phenomena called Kanyava phenomenon. Okay, we'll discuss that later in some other videos. But this is a serious issue, guys. So even if some of you has been like in the working in the industry or any other industry, it's your responsibility. So make sure that people are following the basic hygienic principles so that this kind of outbreaks are not happening, and we should stop this kind of unnecessary deaths, as I want to say. Then viral infections, it basically caused because of the virus, and it may depends on the virus to virus 3 to 6 weeks. And see, virus cannot multiply or any other things until it come in contact with the human cells, right? So that's why human shellfish water is acting as a carrier. Fecal contamination foods it might present, it both enteric and cause both enteric and non-gastric disease. Now let's come to toxico infections. First is Aeromonas, and what is the toxin? It can produce cytotoxin and enterotoxin, and the disease is actually aerolysis. And Aeromonas hydrophila is a bacteria which can cause travel diarrhea. See, this is travel diarrhea because most of the normal people will be like consuming this routinely, so they will be like immune to this specific disease. But if you are going it in a specific day, like if you are going for a trip or something, and if you are new to this disease, then it can cause diarrhea to you. That's why it's called as travel diarrhea. Along with this Aeromonas, we can also say E.COLI, toxigenic E.COLI and pathogenic E.COLI is considered as a travel diarrhea. Okay, so this is a quick example of what is a travel diarrhea. Okay, 24 hours 10 power 4 to 10 power 10 viable cells are needed. And we see most of the like bacteria here is needed large number of viable bacteria. I have told you why it is mentioned large number of viable. Here you get 10 power 4, 10 power 6, 10 power 6 to 9, 10 power 10. Because why? Because see, in order to reach the intestine, it has to have large number of bacteria, right? I have mentioned it before. That's why it contains large number of viable bacteria. So most of them will cause enteric diseases only. You can see enteric. It's almost all the diseases are enteric only, related to intestine disease. So Bacillus diarrhea is caused by Bacillus cereus. There are two types, short and long. In short, it costs from 1 to 6 hours. In long, it is 8 to 16. It's in the seafood, meat and poultry, abdominal pain and diarrhea. See, this diarrhea is also caused from irregular heating. If you are making some kind of fried rice and all, then this bacteria can affect. Then we have a Clostridium perfringens. It's a gastroenteritis. It is a toxic infection. I told you before, right? So it 10 power 6 to 7, 8 to 24 hours. It causes it will be transferred from intestinal tract of human soil or sweater. Sweater is also mentioned in the book. I don't know why, so I include it. To the cooked meat, beans, leftovers, gravy and cafeteria jam. See, Clostridium perfringens is called as cafeteria jam because mostly this disease is caused due to the leftover gravy, because it will act that as a medium to grow the bacteria. Okay, Pelsimonas shigellocysts is 1 to 2 days 10 power 6 viable cells, more than 10 power 6. Aquatic animals and fishes, environmental species will be there. So fish, shellfish and oysters, and it's also enteric. E.COLI, we have pathogenic and toxigenic. It's a travel diarrhea, and in the toxigenic, it's an enterotoxin, 10 power 6 to 10 power 9, 10 power 8 to 10 power 10, and it's 12 to 36 hours. It will show the symptoms. From human feces to vegetables, milk and salads, and it will cause. Cholera, we have Vibrio cholerae. 1 to 5 days and 10 power 6 viable cells is needed. From man, animal, marine to shellfish, salads and water, it's an enteric disease. Okay, intoxication. I haven't mentioned viable cells here. Why? Because we don't need a viable cell. We just need a toxin. The first is Bacillus cereus emetic. See, we have seen Bacillus cereus diarrhea. It is a Bacillus cereus emetic toxin. So it will produce a toxin. Okay, that toxin is actually similar to the MSG's emetic syndrome, Chinese restaurant syndrome. It will be appeared in 1 to 6 hours as symptoms. And from soil to cooked rice, fried rice, puddings. I told you fried rice, right? Because of this point only. I might have confused it. But from soil to cooked rice, fried rice, puddings and sauces, flours and pasta, the Bacillus cereus can be affected from sauces, flours and pasta. But if you are considering baked foods, creams and cheese and ice cream, it is Staphylococcus aureus. If it is pasta, flours and sauces, ingredients, then it's Bacillus cereus. If it is a final product, cream, bakery, cheese and ice cream, it's Staphylococcus aureus. Use this as a differentiator, okay? And both can cause diarrhea, watery diarrhea, diarrhea only, dehydration and diarrhea. And botulism is nervous disorder. Basically 12 to 48 hours you can identify it. Soil, bacteria, soil, birds, fish and animals too. See, one more things apart from this disorders, you can cause blindness from the Clostridium botulinum. Okay, that is actually related with a nervous disorder only, blindness also, but it might ask in the question. So canned foods, garlic in oil, vacuum active foods, we can see this specific bacteria. Next we are going to see the Staphylococcus aureus, 0 to 5 to 8 hours or 1 to 6 hours. There are two types. One is it will come very early or a little bit later than that. Basically from the throat, skin, nose and pimple of humans to the custard, cream, bakery and cream cheese. Why? Because by touching this type of parts in the body and touching the bakery, we are transferring the disease. That's why it is mostly associated with the kind of food items which we use hand mostly. Okay, this is a simple examination. That's why hygiene is really important. Now we are going to talk about the mycotoxin, which is a mold toxin. There are 14 types of toxin which is carcinogenic. In that one, the important one is aflatoxin. It's come from the major aflatoxin is come from the Aspergillus flavus. Aflatoxin means AF toxin. Afla means Aspergillus fla toxin, AF toxin. Okay, we can say AF toxin, AFB1, AFB2, AFB3 and all. So the B1, G1, G2 and M2 is the color which it will produce. B1, B2 is blue, G1 is green, G2 is green to blue, M1 is blue violet, M2 is violet. And if you are taking the B1 toxin, if you are hydroxylate it, you will get M1 toxin. That is present in the milk, urine and feces. In toxicity point of view, B1, M1, G1 is greater than B2, M2, G2. It's the order. This toxin is polyketide secondary metabolite. Okay, and you can see the growth of Aspergillus flavus in 33 degrees Celsius. Optimum temperature is 24 to 28. pH is actually 5. From soil to air, it will transfer from cereal grains to milk to apple to nuts. It's a non-entry. Let me explain how it will transfer. So in field, mold will be affected. In storage, mold will be affected. In damaged grains, mold will be affected. And this mold is produced at a mycotoxin. Obviously, we are not going to process here, right? Now, this from the mold to feed, from the storage to ingredients, from the damaged grain to foods, here mycotoxin is present. You cannot eliminate during this process. You have to eliminate it here. You cannot eliminate it here. You have to eliminate it here. That's why it is a carryover toxin to the food. Okay. Now you have understood how the mycotoxin is present in food. There is one more disease which I am going to explain. This is ergotoxin, ergotism. Okay, black fungi kind of thing. So it is actually present in the rye and bajra, and the toxin is produced by Calceps purpura. And it need a condition of high moisture and 12 to 40 degrees Celsius. So keeping it low moisture is the way. Okay, and the toxin is actually purple brown color toxin, alkaloid ergot, and it called as ergotamine. Okay, symptoms is actually hallucination, confusion and gangrene. Gangrene is the important symptoms. What is a narcotic gangrene? Okay, it means it is a burning sensation, burning fire like sensation. That's it. That is ergotism. I have given all the other mycotoxins here and moles here and toxicities are here. You can read it. I am going to talk about the FSSAI standards only right now. So we have total aflatoxin, B1, M1, ochratoxin, poutine and deoxynivalenol. Okay, so yeah, you have to study each and every type and what is its limits. It's easy only. It is mostly repeating. As you can see, most of the nuts is 15, oil seeds is 15, pulses is 15, only the spice mix is 30. and any other food is 20. Okay, and fix is 10. So you can differentiate it by like mixing, for example, nut, cereals, all are 15 total aflatoxin. 10 is actually figs, and 30 is spices, and 20 is all other food. Okay, so simply by dividing it, you can easily study this, right? So go and study in this way. So next is aflatoxin B1. As you can see, it is in spices also same. It is 15, somewhat higher here. Like here 30, it's 15. In aflatoxin M1 we have milk, skim milk, whole milk. Whole milk it is 4, milk, normal milk it's 0.5, skim milk it's 6. Ochratoxin it's present in wheat, right, and barley it's 20, botulin is 50. Both the apple juice and apple juice as an ingredient is 50. In wheat it is 100. Deoxyvanillin. Okay. Now I am going to talk about the NOTS, naturally occurring toxic substances. Here the toxin is actually naturally present, and how this toxin is affected is depend on the person's sensitivity, concentration and amount of substance. Then the toxin is differ from fungal toxin, plant toxin and marine toxin. And in fungal toxin I have talked about mushroom and like death cap mushroom. So it can be lethal for adult because it can affect the gastrointestinal. And hepatotoxicity is a toxin which can kill our body because of the mushroom toxin. In plant toxin we have many toxins are present, such as hydrogen cyanide, fucomarins, glyalkalides, lecithins and oxalic acid. For example, let's take an example of hydrogen cyanide. You might have heard this word in the it present in the cherries, pits and apples, right? Cherries, pits and apricots and apples. Obviously, yeah, it's present, and it is a bitter in taste. But it's also present in the cassava and bamboo shoots. Okay, so unprocessed cassava and bamboo shoots, this hydrogen cyanide is present. It also known as prussic acid. See, apart from this, there is a specific cyanohydrins is there. It is mandolonitrile and amyloglidin. That will slowly release the HCN. It will slowly releases the HCN. Okay, so unlike directly releasing HCN, it will produce these two and that will release HCN. It is also really present in the almond oil. Okay, in furocoumarins, it is present in the parsnip and also carrot, parsleys and all. So you can easily relate it, and it can cause allergic reactions and some problems. Okay, and it can also cause GI like gastrointestinal problems. So you have to peel it every time and cook before consumption. But it's different from glyalkalides, because in glyalkalides you have to peel, because it's basically because of the sprouts and the like green patches in the potato. You cannot cook and kill this bacteria, kill the specific like glyalkalides. That's why it cannot cooked like here like furocoumarins. You have to peel it and cut off it. Okay, that is glyalkalides. Lecithins is actually you can easily destroy it by soaking in water and leaching it, followed by boiling. And example is green kidney beans. And there are many GI related problems related to this. Oxalic acid is present in rhubarb. If you are using rhubarb from kind of baking pies or other dessert, you have to make sure that it is not too toxic, because the leaves are too toxic. You cannot use the leaves. Stems you can use it, leaves you cannot use it. Remember it. This oxalic toxicity can cause neurological, cardiac, respiratory and GI effects. Then cucurbitaceans. Cucurbitaceans is actually present in the zucchini or courgette, in which we say in USA. So courgette or zucchini, it's present. The cucurbitaceans is present. It's actually it's used in a savory dish because it has a savory flavor to it, and it can cause some GI problems. In marine toxin, the three important toxins is mercury toxin, cicuterra toxin and scrombead poisoning. Scrombead poisoning is actually histamine poisoning. Cicuterra poisoning is the poison which does not have a cure right now, and the cicuterra poisoning is actually happening because of the microalgae. Okay, if any fish is consuming microalgae, then you will have this cicuterra poisoning. Okay, and the symptoms is actually subside slowly, and treatment is actually symptomatic. Yeah, which symptom you have to treat accordingly. Mercury poisoning is because of the accumulation of mercury in the body of the fish. It actually feeding the small, it's actually because of feeding the direct absorption of the small fish without like digesting the direct absorption of small fish. For example, the big fish like shark and large sea fishes, it will like swallow a large number of small fish. In that small fish, along with that, the mercury will also come and deposit in the body. So the young women and children cannot consume this, okay, in the early days. Next, Shigitara poisoning, I have already discussed it. It causes nausea, vomiting, and it based on the symptoms you have to treat it. And it may be present in black grouper, black fin snapper, and then king mackerel and all. Scrum-bit poisoning is actually histamine poisoning. See, FSSAI has a list of fishes under histamine poisoning like mackerel, herring, sardine yellow. I have mentioned few. There are list of fishes is there. You can go and see what kind of fish is actually accumulate the histamine poisoning. It's actually because of the inadequate storage and temperature control of fish. If you are catching the fish, you have to store it and keep it in the temperature control. You can clearly see after consuming 30 to 1 hour, you can clearly see if you have a toxin, if you have symptoms because of the vomiting, nausea and other things. Important symptoms is tingling in the mouth and dizziness and headache and blurred vision. Okay, followed by the fish oil poisoning. It is very rare and it's in the fish like escalar and oil fish will be there. You have a diarrhea and the diarrhea will be oily consistency. That's how you need to understand that this fish poisoning is there. The FSSAI has given for NOTS, naturally occurring toxic substances. FSSAI has given 4 substances as a primary substance. Agaric acid means the acid which is present in the fungi and the ppm is 100 ppm. Hydrocyanic acid, we know it's in the black cherry, apples. I have given the scientific names if suppose they have asked, and the ppm is 5 ppm. Hyperzine is actually present in the flower Hyperzium. Okay, it's a St. John wort flower you can see here. Sometimes we use the extract of this flower, because of that the hyperzine might present. Then saffrole. Saffrole is actually present in black pepper, cinnamon, nutmeg and all other essential oils and all, but it majorly present in the sassafras. So this is the sassafras tea which we can see leaf. You can identify by the looks of the tree, and this saffrole is present in the sassafras tree, and this can also be leads to a lot of other problems. So that's why 10 ppm is the range given by the FSSAI. So these are the important maximum limits of the substances here mentioned here. Okay, that's it. We have concluded the foodborne diseases and the foodborne pathogens. If you like the video, please make sure to subscribe the channel and give a like to this video and share it with your friends, so that they can enjoy the video and they can learn the food technology for any other food related examinations. You can download the PDF, but it will be available only for fewer amount of time, so make sure to join right now to the Telegram channel to download this PDF. And you can also join the Telegram quiz. Okay, every day 7 PM we'll conduct the quiz, except Sunday. Sundays will be like a repetition of the whole week quiz. Okay, so there will be no new quiz in Sunday, but we'll conduct the quizzes also, like the old quizzes. But all the 7 days from Monday to Friday 7 PM you can attend new quizzes from our Telegram channel Food Catalyst. This is Venkadesh and we have done with the food microorganisms. That's it. Bye.